Welcome. Good morning. Thank you for joining us at today's seminar. We're going to get started in just a moment. If you're joining us on Zoom, please remember to use the Zoom Q&A to ask questions at any point during the seminar today. And if you're joining us on YouTube, please use the YouTube chat. So we're now right at 10:30 Pacific time. So I'm going to hand it over to Farzaneh to get us started.
Hello, everyone. Welcome to another presentation in our distinguished seminar series. As you might know, in these seminars, we feature outstanding scientists based on the impact of their interdisciplinary research to the neuroscience community. Today, we're featuring a talk by Andrea Gomez. She's an assistant professor at the Department of Molecular and Cellular Biology and the Helen Wells Neuroscience Institute at UC Berkeley. Andrea's research is motivated by a genetic paradox, which is the lack of correlation between the number of protein coding genes and the brain complexity across species. One way to expand variation in cellular regulation is through RNA diversity, and that's what Andrea is interested in. In particular, she uses a variety of methods, such as neurophysiology, behavior, synaptic biology, and molecular genetics to study the link between RNA diversity and synaptic formation and function. Let's talk a little bit on Andrea's educational background. She did her bachelor's in Colorado State University in biological sciences. She finished her bachelor's in 2006 and then started her PhD at NYU in developmental genetics with Stephen Burden. She studied the development of the neuromuscular junction, and her PhD work led to the discovery of a protein that mediates the interaction between motor neurons and muscle fibers. Andrea finished her PhD in 2014 and then moved to University of Basel in Switzerland to do her postdoc with Peter Schaeffel. There she studied the molecular principles of synapse formation and function. Her postdoc work demonstrated how RNA splicing is essential in the development and function of synapses in the mouse hippocampus. Andrea finished her postdoc in 2019, and then last year she opened her lab at UC Berkeley to delve into the mechanisms that specify synaptic properties. Welcome, Andrea. Please go ahead and start your talk. Thank you. You're
muted.
Is everything, I'm still muted?
Nope, you're back now.
Okay, great. Alrighty. Thank you, Farzaneh, for that great introduction, and also for the invitation to this distinguished seminar series. I'm really excited to share my work and the work of my group. So, as mentioned, a little bit about me. I'm a trained developmental geneticist, and I suppose you could say that my scientific quest is to understand how we became so complex. And more specifically, what mechanisms provide the substrate for complexity to arise in the first place, and to link these complexity generating mechanisms to the way fluctuating ionic current is patterned in single neurons to neural circuits. And this quest to understand these questions has led me to alternative RNA splicing. So I would like to spend a little time in the beginning of my talk to provide a framework for how we are thinking about questions in my lab, which is to understand the organizing signals of the brain. What is the nature of these signals and how do they organize? And while we know the genome is the locus of the instructive cues, the genome itself cannot directly facilitate organization. Instead, it relies on RNA to coordinate the instructive cues to alter neural circuit function. So do these signals, or rather, how do these signals organize the connections between neurons or synapses? Really, what information in our genome, in our DNA, instruct for the connections of neural activity that's responsible for our thoughts, our memories, our feelings, and our behavior? Now, how do we even begin to ask how an overwhelmingly complex structure such as the brain is organized? So for the organizing signals, as the title suggests, I look to RNA. And if there is one take home for you to remember today, is that the complexity of our brain is matched by the complexity of RNA. My lab focuses on our RNA alternative splicing. And again, the diversity and how that diversity is generated in the brain, and we link this diversity to synaptic function. To achieve this, we combine genetic strategies, the latest deep sequencing technologies, and we match these strategies with electrophysiology and functional imaging to understand how RNA transmits the cues to the millisecond computations in the brain. So what is this image you've been looking at? Well, this is an entire mouse brain where we have genetically labeled a specific region called the hippocampus, a region that is crucial for memory and spatial navigation. Now here is another view of the hippocampus. Now, when we think about network activity propagating through this circuit, and it does so in this general direction, we know that it is not continuous. It is in fact interrupted as activity passes from neuron to neuron to neuron throughout the circuit. Inputs summate, trigger an action potential, and repeat. Now, the fidelity of this transfer relies profoundly on synapses, or the connections between these neurons. And this is why I like synapses, or why I am so interested in the properties of synapses, because they have a profound effect in shaping the way that neuronal activity is propagating throughout this network. But what are the cellular mechanisms for organizing these properties? Well, for one, we believe that a significant amount of the network is predetermined. And we think that unique transcriptional programs that are activated in distinct cell types direct both the assembly as well as the maintenance of a network. And it is my main goal to decode what molecular programs are contained in these cell type transcriptional repertoires that contribute to synaptic properties. Now, if we zoom in on a single pyramidal neuron, we can appreciate the many different levels of stereotype synaptic specificity. We can appreciate that it is targeted by various classes of neurons, and cell type specification enables all these inputs to appropriately identify and synapse onto its target with subcellular precision. Now, each of these connections also have different synaptic properties. For an example, one may have a specific neurotransmitter that mediates excitatory or inhibitory currents. Another may target special components to the presynaptic terminal that tunes the probability of synaptic vesicle fusion, resulting in a facilitating or a depressing synapse. One synapse may have special plasticity features such that when triggered by a strong or specific stimulus, increases the number of neurotransmitter receptors in the postsynaptic membrane, thus strengthening synaptic transmission. So how are these properties established? Well, we believe that these occur through transsynaptic signaling modules, or transsynaptic linkers, each expressed by the presynaptic cell and the postsynaptic cell, that when bound, insist together the components of the presynaptic release machinery, as well as signal to organize neurotransmitter receptors in the postsynapse. And we define this transsynaptic signaling module as the minimum unit that can facilitate this bidirectional activity. Now, these modules, which we envision as senders and receivers, transmit organizing signals that depend on the identity of the transsynaptic linker and the components that they recruit. And here we are depicting the same sender with different receivers that result in different messages or different instructional cues in these two different cellular contexts. Now, depending on the synaptic context, the brain region, the cell type, the content of these messages act to drive overlapping elements of synaptic organization, such as bidirectional adhesion, presynaptic vesicle recruitment, active zone assembly, or postsynaptic stabilizations of neurotransmitter receptors. And we also envision that these transsynaptic signaling modules, or these sender and receiver pairs, can be mixed in combinatorial ways to generate the diversity of synaptic properties we observe in neural assemblies. For an example, this pair may have a specific neurotransmitter. This one may have special plasticity features or have unique release probabilities. Now, in this model, the only limiting factor for how many different types of synaptic properties that can be achieved is simply the number of available sender and receivers, which brings me to alternative splicing. And I kind of take this analogy from Chin Lo, and to illustrate this point, we have 20,000 genes, mice have 20,000 genes, even C. elegans has about 20,000 genes. However, humans with our 90 billion neurons make about 600 trillion to 2 quadrillion synapses, mice and their 75 million neurons, 500 billion to 2 trillion synapses, and the worm with 302 neurons and 7,000 synapses tells us that the number of genes does not scale with brain complexity, which is why I'm so interested in alternative RNA splicing. So we all know from the central dogma that DNA gets read into RNA and RNA gets read into protein, but before mRNA can be read into protein, it needs to mature. Introns need to be cut out and exons spliced together. And what can happen during this splicing, which is diagrammed here, is alternative choices in exons can be made. And this happens through RNA motifs that and transacting factors, which either promote or suppress the usage of specific exons, which then go on to make distinct mRNA isoforms. Now, relating this back to the synapse, alternative splicing has the potential to vastly expand the coding power or the synaptic proteome, and thus increase the modularity of the synaptic or the transsynaptic signaling modules for these sender and receivers. And with alternative splicing, here we have a mechanism that can expand molecular complexity, expand the coding power of the genome without disrupting the underlying transcriptional programs that have stood the test of time. And interestingly, the highest degree of alternative splicing is in neurons compared to any other tissue in the body. And the highest degree of alternative splicing has been observed in primates, showing that splicing scales with network complexity or with brain complexity. And finally, you may not be surprised that alternative splicing is also highly dysregulated in neurological disorders. And I just want to quickly highlight the large variety of patterns that can be generated by alternative splicing. Here on the top is the most common isoform, the most common form, which is the subject of today's story, which is the inclusion or exclusion of specific exons. But I just want to point out that there are lots of different types of alternative splicing. There's alternative use of promoters, alternative polyadenylation, which generate larger or shorter 3' UTRs. And really, from this, we can appreciate how, from a single gene, alternative splicing can generate a large diversity of mRNA isoforms. Now, to go back to our model of how cell type specific alternative splicing choices are made, the final but most critical aspect of our model is that we hypothesize that there are splicing regulators that are selectively expressed in different cell types that drive unique splicing events that shape synaptic signaling. And so let me introduce you to the RNA binding protein SLM2. And for the reasons I just stated, we became extremely interested in SLM2, which is an RNA binding protein, because we found that SLM2 displays non-overlapping expression in various cell types in the hippocampus. And again, the hippocampus is a highly conserved structure, essential for memory and spatial navigation, just to name a few. So we generated SLM2 knockouts and got motivated that perhaps SLM2 had some function in the CNS because they had a weird behavioral phenotype in that they had an unusual response towards novelty. Now, normally when a wild type mouse is presented with a novel object, it will explore the novel object, here seen in an increased amount of time spent with the novel object. However, our SLM2 deficient animals displayed no interest in the novel object. In fact, they displayed a perseverative behavior in that they preferred the familiar object. Now, we don't believe that this was an anxiety-like feature because when we tested them in different anxiety assays or anxiety-like assays, this one, the elevated plus maze, we can see that this SLM2 deficient animals did not avoid the open arms of a maze, which is thought to be a source or a space of vulnerability. So the loss of this RNA binding protein caused novelty defects in behavior. Are these changes associated with changes in the transcriptome? So determine what, if any, changes this RNA binding protein made. We performed a comprehensive global analysis by whole genome RNA-seq, and using Illumina paired end sequencing of libraries prepared either from wild type or SLM2 knockout hippocampus, we observe highly correlated expression levels of genes comparing wild type and knockout tissue. And this indicates that SLM2, this RNA binding protein, does not tune overall RNA levels. Now, what about splicing? Here is a volcano plot where each dot is an exon, and we can see the statistical differences in included or excluded exons comparing the wild type or the knockout hippocampal tissue. And what we can see is that we only see a handful of exons that are deregulated. Now, this was an incredibly surprising result because most known RNA binding proteins regulate hundreds, if not thousands, of transcripts. And perhaps even more impressively is that the deregulated exons encode for synaptic adhesion molecules, Neurexins 1, 2, and 3, and the syntaxin binding protein, Talmasin 2, all of which localize to the presynaptic terminal. So just a little bit of background on Neurexins. Neurexins are ancient evolutionarily conserved neuron genes. They came about the same time that nervous systems came online, and they are extensively alternatively spliced. Now, with 3 different genes, 6 alternative splice positions, 3 alternative promoters, theoretically can generate 12,000 isoforms. And we and others have confirmed about thousands of these isoforms existing in the mouse brain. Now, when we look at the identity of the splicing changes that we observed in the SLM2 knockouts here in this qPCR, we can see that and again, just this is the recapitulate the volcano plot we saw earlier, in that wild type animals, they have splicing or inclusion and exclusion at this alternative splice site 4, and in approximate equal amounts. Whereas an SLM2 knockout hippocampus, we lose this skipping. And we can see that this shift from the minus isoform or the skipped isoform now includes predominantly this isoforms that contain this exon at the alternative splice site 4. Now, what part of the, or what does this exon encode? Well, this exon encodes for 30 amino acids. And here I've mapped these, this alternative splice site 4 onto the Neurexin protein, and we can see that increasing the length or increasing length of 30 amino acids at this position is poised to increase the length of the protein, which sits at a ligand binding interface to canonical ligands like LRTM and neural ligands. So to test if our splice variants are preferentially binding, distinct postsynaptic or synaptic components, we took an unbiased approach to determine what preferentially bounds to the isoform that contains the exon at the fourth position or lacks the exon at the fourth position. And we immobilized recombinant Neurexin 1 that either contained or lacked, and we ran hippocampal lysate over this column. The column was eluted and sent to mass spec for shotgun analysis, and our analysis identified 21 candidates. Many of them have been well characterized. For an example, LRTM2, a membrane-bound postsynaptic cell adhesion molecule, and target Neurexins preferentially bound to the skipped isoform of Neurexin, whereas cerebellin, another well-known target of Neurexin, preferentially bound to isoforms that contained the exon at alternative splice site 4. We also identified new interactors, chattel, a leucine-rich extracellular matrix protein, which can bind collagen and inhibit the maturation of the extracellular matrix, preferentially bound the skipped Neurexin, whereas complement 3, which has been shown to mediate microglia-mediated synaptic pruning, preferentially binds to the non-splice or the isoform that contains exon at the alternative splice site 4. So now we know which proteins preferentially bind to one isoform over the other. Does the loss of splicing at this site change the affinity of Neurexins to known ligands in our SLM2 knockout animals, where we know that the isoform that skips the alternative splice site at splice 4 is specifically lost? And so for this, we pulled down Neurexins from hippocampal lysates from wild type and SLM2 knockout tissue. And indeed, endogenous Neurexin binding to neuroligin 1 and neuroligin 3 is lost in SLM2 knockouts. So now that we have some idea about potential mechanistic changes at the synapses, does this change the function of the synapses? So first, does splicing lead to structural changes? And first, we saw that disrupted splicing did not affect overall structure of the hippocampus, including the density of spines, which are the primary sites of synapses in CA1. Next, when we measure spontaneous synaptic events from CA1 neurons, knockout neurons were indistinguishable to wild types in both the amplitude as well as the frequency of these miniature events. However, when analyzing the kinetics of these individual events, we did observe a very minor speeding of the spontaneous events, evidenced by an increase in the rise and the decay times of these transients. Now, what this suggests to me is that this may indicate that the composition of the postsynaptic properties are disrupted. And when we look at the components of the postsynaptic receptors, we observe a significant increase in the AMPA receptor subunit GluA1 measured on the surface of hippocampal tissue. And this coincides with an increase in excitatory current that is mediated by AMPA receptors. And here, when we subject a whole cell recorded CA1 neuron to a special plasticity program, which normally strengthens wild type synapses, SLM2 knockouts failed to undergo long-term potentiation, which is a process we know critically depends on AMPA receptor regulation. So just to kind of wrap up this last section, we set out to ask if alternative splicing was important for the specification of synaptic properties. And when we disrupted a splicing regulator by knocking out an RNA binding protein, we found that SLM2 is critical for a selective regulation at the RNA level. It wasn't changing overall upregulation or downregulation of specific genes, but specifically splicing was disrupted. Now, at the level of the synapse, again, a remarkably selective phenotype marked by a change in the level of AMPA receptors, which was coordinate with an increase in excitation. And since Neurexins were targeted by SLM2 mediated splicing, we hypothesized that Neurexin splicing in the presynaptic neuron generates a transsynaptic signal that controls the magnitude of synaptic weight through AMPA receptors. Now, if this hypothesis is correct, then deleting SLM2 only from the presynaptic neuron from the CA3 neurons should produce the same phenotype. And the uniqueness or advantage of this strategy is that this would also rule out the possibility that the phenotype that we are observing is coming indirectly from somewhere else in the circuit that SLM2 is expressed. And finally, to prove that the transsynaptic signal is indeed carried out by neurexins, we restored wild-type repertoires on neurexin isoforms. And we expect that this should restore wild-type synaptic function and behavior. So first, to probe presynaptic function, I made a conditional knockout by specifically deleting SLM2 from the presynaptic cell. To achieve this, I utilized a cell type specific pre-driver that is selectively expressed in CA3 neurons in the hippocampus. So first, to confirm the selectivity of this pre-driver, I crossed this line into a Phlox TD tomato mice and assessed the extent of pre-recombination using 2-photon tomography. And from this, I think you can appreciate just how selective this Cree driver is. Now, crossing this Cree driver into SLM2 Phlox mice, we are now able to generate a targeted deletion of SLM2 from the CA3 neurons. Now, this is likely that these residual SLM2 positive cells are interneurons, but at least we can confirm that we are seeing about an 80% reduction in SLM2 positive cells in the CA3 region. Now, what's happening to the splicing in these SLM2 deficient CA3 neurons? And just to remind you, before we did RNA-seq and assess the splicing pattern in whole hippocampal tissue, but we can't use this method anymore. We now need to selectively look at the transcriptome only from CA3 neurons. And so to do this, we use the ribotag technique, whereby ribosomes get selectively labeled with an HA tag in a Cree-dependent manner. We can then pull down these HA tag ribosomes, which should be only tagged in CA3 neurons, and assess the RNAs that are attached. And if we look at wild-type CA3 neurons, Neurexin splicing is almost entirely excluded at this fourth position. You can see that the predominant isoform here is the skipped version. On the other hand, selectively deleting SLM2 from CA3 neurons results in a loss of alternative splicing at the fourth position. And now these transcripts shift up, and which refer to the transcripts that are containing this exon. Now, because of the highly selective expression of the Cree driver, we can directly test the effect of Neurexin splicing in the presynaptic cell onto downstream targets. Now remember, I'm only deleting SLM2 from CA3 neurons, so the CA1 neurons that we are recording from are wild type. And when we measure the magnitude of synaptic strength measured on wild type CA1 neurons that are contacted by mutant CA3 axons, indeed, we see an elevation in excitation. And this confirms that splicing in the presynaptic neuron instructs for postsynaptic strength. Now, I've shown you what changes to synaptic current looks like, but there are other types of currents, like the voltage gated channels, that contribute to a spike, or the ones that determine the resting membrane potential are just important for neuronal integration. So first, because it is known that the changes in strength of synaptic input can induce an adaptation in these intrinsic electrical properties, we first wanted to assess the response of wild-type CA1 neurons that are contacted by CA3 mutant axons in the firing frequency. And recall, these wild-type CA1 neurons have elevated synaptic excitation. And when we do a input-output curve on the firing frequency, we see somewhat of an expected result in that the response of these SLM2, or rather these CA1 neurons, is to reduce the firing frequency of these of these cells that are receiving all this extrasynaptic excitation. Now, to identify the potential contributors to why would we see a change in these firing frequency, we looked to the HCN channels for their ability to change the membrane capacitance and resistance of the cell, which could alter the firing frequencies. And indeed, wild type CA1 neurons that are receiving all this excitation have an adaptation in that they receive increased currents that are mediating through these HCN channels. Now, this was selective because when we looked at the action potentials or the active properties of these CA1 pyramidal neurons, we see no difference in the spike threshold, the height, the spike width, or the after hyperpolarization, suggesting that at least the output, or once the action potential gets triggered, there doesn't seem to be a change due to this extrasynaptic excitation. So just to kind of summarize this last set of data, we found that presynaptic control, or rather alternative splicing in the presynaptic neuron, can transsynaptically control intrinsic properties in the postsynaptic neuron. And this was observed through increased IH currents, decreased firing frequency, faster time constances, and increased capacitance. Now, going back to the behavior of these animals, does this extra excitation from CA3 to CA1 alter any behavior in the animals now that are selectively deleted in the hippocampus? And we looked at this open field paradigm again, and while distance, the number of center crossings, and the time in the center were indistinguishable to wild type, we did observe this modest but significant decrease in the velocity of these animals exploring this open field paradigm. So just to kind of circle back, these results argue that specifically SLN2 in the presynaptic cell generates a transsynaptic signal that is responsible for synaptic phenotype in the target CA1 pyramidal neurons. Now, we believe that this is mediated by neurexins, but we cannot confirm this. This is an RNA binding protein. It could target other genes that we were unable to detect in our RNA-seq. So to test this hypothesis, we need to restore the correct neurexin splicing in the presynaptic neuron. Now, I hope you can bear with me, but this does involve a little bit of somewhat complicated genetics. And our strategy was to effectively mimic the splicing that SLN2 does. So just to recall, in wild type neurons, the predominant isoform of neurexins in CA3 neurons are this skipped isoform. Now, to restore that, we did a genetic mimicry whereby we put flocked sites around the alternative splice site 4 from Neurexin 2 and 3. We also got a Neurexin 2 allele from the Sudoff lab, which is similar to our same parameters in that there's flock sites flanking this alternative splice site 4, so that we could effectively mimic the splicing that SLN2 does by genetically cutting it out, by deleting it with CRE. And so to generate a rescue mouse, what we did is we crossed a CRE expressed in CA3 neurons in SLN2 flox animals with these neurexins that have this flock site around. And so our rescue animal would contain CRE, SLN2 flock, so we would delete delete the RNA binding protein, and then restore or mimic the splicing that SLN2 does by deleting the AS4 allele of Neurexin 1, 2, and 3. And when we measure the magnitude of synaptic strength of wild type neurons that again are contact by mutant CA3 neurons, again, here on this pink, we see an elevation in synaptic excitation. However, our rescue, so putting back or mimicking the splicing that Neurexin does of Neurexin 1, 2, and 3 in CA3 neurons, restores the magnitude of excitation. Actually, it overcorrects, and that it reduces the synaptic excitation even below wild type levels. Now, we're not exactly sure why this overcorrection is happening. However, this does confirm that presynaptic, or rather splicing in the presynaptic neuron is instructing for postsynaptic strength. And so now, does this change the other phenotypes that we observed? This elevation in AMPA receptor, or these transsynaptic interactions with postsynaptic adhesion molecules. And again, just to reiterate, now this is a single Neurexin rescue. We targeted Neurexin 1 using the same strategies I described before. And a single exon of Neurexin 1 was able to restore the interaction of Neurexin with the postsynaptic ligands, neuroligin 1 and neuroligin 3. And restoring this single exon in Neurexin 1 was also sufficient to restore the elevated AMPA receptors back down to wild type levels. And further, the plasticity defect that was caused by SLM2 deficiency was also partially restored by the single Neurexin 1 rescue. And finally, the weird novelty behavior in SLM2 knockout mice was also restored to wild type levels. And again, the novel object animal, the novel object task, these animals now spend kind of the expected elevated time with the novel object when it was presented in the novel object task. We also, this rescue also applied to the other behaviors that we saw in the open field. In the full knockout, we see an elevated velocity in the maze. This was restored to wild type levels in our rescue. And this, this kind of weird marble bearing task, which was reduced in our SLM2 knockouts, was also restored to wild type levels. Now, in this particular case, we don't necessarily claim that these behavioral phenotypes are driven only by hippocampal dysfunction. However, our results do support that the function of this RNA binding protein, the function of SLM2, is indeed regulating a limited number of alternative exons in the Neurexin gene family. So just to wrap up, as I mentioned in the beginning, I set out to identify molecular mechanisms that specify synaptic properties. And I shared my view of how a synaptic adhesive code may manifest in the central nervous system. But, we're still left with a kind of an elephant in the room, which is how robust is this code from mice to humans? How is the code manifested when you change the number of neurons by an order of magnitude from mice to humans? And this is why I'm interested in alternative splicing, because this mechanism can increase the coding power of the genome without disrupting the underlying evolutionarily conserved gene expression patterns and overlay another level of complexity. But for this to make sense for the brain, which is a highly heterogeneous system, increasing molecular complexity, it needs to happen in selective ways. And as I've shown you today, we discovered an alternative splicing program that is devoted to the splicing of synaptic adhesion molecules. And when this was disrupted, not only did it alter behavior, but it also disrupted excitation and synaptic plasticity in the hippocampus. Importantly, by selectively disrupting splicing in a subset of neurons, we were able to demonstrate that alternative splicing can shape neural circuitry in cell type specific ways. And the last point is especially exciting because considering that 95%, pretty much all genes, or multi-exon genes rather, are indeed alternatively spliced. So what other cell type specific mechanisms are out there? Can different combinations of RNA binding proteins encode other aspects of synaptic function? Now, our discovery that alternative splicing can shape neural circuitry by selectively modifying synaptic properties is just one study in a growing body of evidence that has been shown by us and others that splicing is a powerful mechanism for network organization and function. And this is especially exciting considering that, despite having comparable number of genes amongst vertebrates, the extent of splicing has been expanded in species with more complex nervous systems, such as mammals. And again, the highest degree of complexity has been observed in primates, which positions alternative splicing as a potential driver for the emergence of complex networks. Now, from the scope of synaptic integration, small changes to synaptic properties, like we observed in SLM2 mutants, would have a large effect on the information flow to a neuron engaged in ongoing network activity. And in knowing this, it may not be so difficult to then imagine, how circuits may be changed in neurological disorders where we know synaptic dysfunction occurs. And this last point is especially relevant as disruptions to alternative splicing has been implicated in disorders ranging from autism to spinal muscular atrophy. Now, finally, understanding how splice codes are distributed across these different cell types, how are they overlaid, we can start to derive some cellular rules for how these splice codes affect neural integration and computation. So now, looking forward, what are the types of questions that my lab is pursuing? Now, the data that I presented today was how splicing regulates synaptic function in a single pathway from the CA3 neurons onto CA1 neurons. But CA1 neurons, they receive information from all over the brain and other streams of information like the entorhinal cortex or LA3 pyramidal neurons. And in fact, the timing of these inputs and how they arrive onto CA1 neuron is essential for plasticity in the network. It is essential for how these neurons are integrating information from different regions. Now, what would we predict if we increase synaptic strength on a single channel? Which is essentially what's happening in SLM2 deficiency. Well, we would predict that if we increase synaptic strength as in one channel, we would predict that the time window for summating inputs from other regions of these CA3 neurons would be effectively shortened. Or another way to think about it is that the bandwidth of CA1 neurons is effectively decreased because each of these synaptic potentials have a greater magnitude and are more likely to trigger or get to that sweet spot of the action potential and trigger it before other inputs can come in. Now, the information from the entorhinal cortex, that information still needs to be processed, but now we are effectively biasing the information that's being fed in from CA3 neurons. And I'm really interested in understanding these types of relationships at this level, because understanding the role that autism risk alleles like Neurexins have on regulating the timing of inputs that converge onto a single neuron may be essential to finding a cellular correlate for the impaired ability of some individuals with autism that have issues with processing contextual information, such as multisensory processing. Further, another avenue that we are really interested in applying this concept of how these splicing codes are arrayed onto neural networks is to test how these splice codes are operating in different neural circuit contexts. Now, SLM2 is also expressed in the cortex, but considering pyramidal neurons in the hippocampus, it may not be such a far shoot to expect that they would behave differently in the cortex. And so to really test this idea that these splice codes are conserved and can operate in different cellular contexts, we are testing these concepts, looking in the enteric nervous systems. We're looking in the gut. And here you can see that these SLN2 positive nuclei are indeed expressed in the neurons of the gut. And finally, the last bit, or last direction of the lab is to kind of expand on a question of what type of plasticity mechanisms are encoded through alternative splicing events. And for this is a group of projects in my lab, which is collectively termed as CRISPR-Sci, which are psychedelic-inspired plasticity therapeutics. We are really interested in the large-scale plasticity that is induced by psychedelics for multiple reasons. Is that there's millennia use of psychedelics to open pathways for cognitive connections and pathways to healing. And these kind of large-scale events have a very selective output. I mean, a lot of things can induce plasticity, but the positive outcomes of the psychedelic-induced plasticity is what we're really interested in. So we have two objectives with the CRISPR-Sci project, which is to one, ask what can psychedelics teach us about plasticity? And the second is to mimic the psychedelic-induced plasticity. And I think with that, I will wrap up and have some time for questions, but I would like to acknowledge my group here at Berkeley. We're new, we started through the pandemic, but we are really excited to answer these questions. I especially want to give a big shout out to Lisa Traumuller, who is a grad student who is now a postdoc in Mike Greenberg's lab, who did a lot of the work with some of the first part of the talk, who was also an excellent teammate in doing this work. Also Yuki Obata, who is starting his lab at UT Southwestern, who was responsible for looking at SLN2 in the enteric nervous system. He is the enteric nervous system, guru, in my opinion. So with that, again, thank you so much for this opportunity to share my lab's work, and I'm very happy to take any questions.
Thank you so much, Andrea. That was a very exciting talk. There is one question from Forrest on Q&A. Do you have access to that, or should I read it for you?
Oh yeah, okay, I'll read it. So,
okay, awesome.
I was compelled by the model presented at the start of the talk. It seems that to realize the complexity of combinatorial programs, there needs to be many more proteins like SLN2 expressed in combinatorial ways across different cell types. What are the prospects for identifying broad or palette of proteins affecting splicing with the same levels of specificity on synaptic physiology? Or do I think the profiling of the variably spliced directly is more likely to be easier, the easier path for identifying these combinatorial patterns and modules? So I think, I urge you to take a look at the Ferlanis et al. Paper published in 2019 from the Scheifele group, who did a really great survey of RNA binding proteins expressed in different cell types in the cortex and the hippocampus. And what was pretty remarkable is that the splice, not necessarily the RNA binding protein themselves, but splicing were actually 4 to 6 orders, or I guess, not orders of magnitude, but 4 to 6 more times likelihood to predict GO models in these different cell types. Like, for an example, comparing CA3 to CA1, or comparing CAMK2 in the hippocampus to CAMK2 in the cortex. Now we suspect that these splicing, these RNA binding proteins are overlapped, and we know that they are indeed overlapped. But making that connection, the splicing patterns plus the RNA binding proteins is a pretty big task. And I would add another level into that, which we know the epigenome also can affect splicing. The way that it can do so is by, if you think about methylating events on the epigenome, can slow polymerase. And this slowing of polymerase in these kind of hotspot regions would effectively allow more time for these RNA binding proteins, these splicing factors to accumulate and make these decisions. So that's how I think regarding your first question. Now, the second question, profiling the variably spike is directly to be the easier path. Yeah, I think I guess that's kind of my approach, is to look at the splice RNA. I think linking the RNA binding proteins plus the epigenetic factors, there's definitely a combinatorial, code that's happening there. But I think it's kind of difficult to kind of generate kind of a large scale. I know the Tilger lab has done like a really good job at doing something very similar to this, but functionally, these are kind of more, I would say, like transcriptomic and computational studies. But the function, like proving that these networks, these RNA binding networks are directly, driving these selective splicing events is a greater task. And I think that, I would say that's my approach. I think I'll answer the next question by Irina Yavorska. My apologies if I didn't pronounce your name correctly. If not anxiety driven, what do I think the causes of the animal's behavior with the novel objects in SLM2 knockout mice? Yeah, I have frankly not a good idea. But if I were to speculate, I think that the timing is really critical. So going back to the kind of the model or the idea that I was telling you about, like this shortening of the bandwidth of these CA1 neurons from integrating, these processes from other parts of the brain, I think it is probably biasing, I guess the relationship of an animal to novelty. I know that's not a very satisfying question, but we have all the data that I presented is slice electrophysiology. I think to kind of get at that question, we would need to do some in vivo, either electrode work or imaging work, which both are challenging in themselves. Okay, so Katie Xiao asks, are the SLM2 regulated splicing events also developmentally regulated? Yes. What are the implications in newborn neurons compared to adult neurons? Okay, so so SLM2 stands for SAM68-like molecule 2. It's in a family of 3 genes. The first one is identified with SAM68. Now, what's interesting about SAM68, it is activity dependent in that, like, if neurons are hit with potassium chloride in culture, these cells will induce some splicing of neurexins. But SLM2 is actually, we think, kind of like a terminally differentiated gene in that once it's on the cell, then we think that it's just biasing cell in that way. And these go on in embryonic brains. So we think that these splicing choices are dictated early on. And what are the implications in newborn neurons compared to adult neurons? I would anticipate just kind of establishing like the biases of networks. If we think that the synaptic strength is kind of important for like a flow of network activity, knowing that the network has all its internal set point for needing to kind of integrate all these streams of information, I think that I think that the best evidence is comparing the knockout animals, which were deleting SLM2 early on, to the cell type specific CA3 knockouts. Actually, this development of question I was hoping to answer with the CA3 CRE, but unfortunately for that question, the CRE doesn't really turn on until about P14, and I actually don't see SLM2 going down until about 5 weeks. Now, the 5-week time point is now relating to the synaptic phenotype is also delayed by another 3 weeks. And it is important to think or to remember that, we're knocking down an SLN2 RNA binding protein. Our SLN2 RNA has to go down. And then only once all the SLN2 protein and all the SLN2 RNA is gone, then can Neurexin splicing be changed. So then we have the isoforms that are already there. Those need to go down. And now the new transcripts of splicing that has to then, and we think that's about a 3-week time point, which I think is also an interesting separate question to kind of showing how the robustness of these complexity generating mechanisms. But yeah, sorry, that was a long-winded answer to your question. I hope that I answered it. Silke, sorry, I'm really sorry for butchering your name. My apologies. I mentioned that splicing increases with brain complexity. What quantitative measures of splicing have been born to support this statement? I'm especially curious about comparisons between mice and primates. So just to answer that question, the Glencoe lab actually had this really amazing study by a few years ago, really comparing the degree or the extent of splicing across various animals. I think there was probably about 6 different species compared. And so at this point, we still need to test the function, but right now what we have is a correlation. Is with the increased alternative splicing is correlated with network complexity. Another study by Tilger group also showed some really interesting complexity in single cell, I think, studies. But yeah, I would say that it's on, I guess, the remaining questions is to kind of show our link functionally, like going beyond this, this correlation. And I think in part, showing that splicing in our context, that what I showed today is that it can have profound effects on the way that the network is functioning, right? Novelty behavior is critical for animal survival. So just by deleting one exon, by essentially reducing these spliced and non-spliced isoforms to single unspliced isoform, we can dramatically affect behavior. And I think that this type of linking function to these molecular mechanisms will be critical to really fully give a satisfying answer to this correlation. Could it be that preferentially the numbers of neurons and not the splicing complexity between these mammalian affects brain? I mean, splicing affects the differentiation of neurons in development. Splicing is especially enriched in development, and it is critical for making choices. But so what is even allowing for the number of neurons to be increased? We know that these states of transiently amplifying neurons, like these maintenance states, are critical. But what's allowing for these generations and these expansions? For allowing a lot of the diversity. One interesting note about alternative splicing in correlation to just general transcriptional rules, that genes, or rather the length of neurons the length of genes in neurons are on average longer compared to any other cell in our body. And this has kind of been the hypothesis for why there's a larger degree of alternative splicing, because if you have longer genes, you have more exons, and you just have a higher chance of having an alternative splicing event. Okay, that's all the questions I've seen.
Thank you, Andrea. I think it's 11:30 and we can close up. Thank you so much for attending.
Thank you.
Have a great day.
You too.
Thank you. Thank you, everyone. This is our final distinguished seminar of 2021. Please come back and join us again in 2022.